it's the Public Health Alliance for Genomic Epidemiology, and it came out of a series of meetings towards the end of 2019, back when we were able to meet each other, for a space for practitioners of largely public health bioinformatics focus on pathogens to find each other and work on developing standards and best practices, and also developing ways to bolster the global workforce of public health bioinformaticians. It's got a website, phage.org, phage with a 4, with a 4. And we have various working groups working on aspects as wide-ranging as infrastructure, which I co-chair, the infrastructure working group. And then we are looking at pipelines, so coming up with best practices on pipelines for data analysis, and you can look forward to a publication on SARS-CoV-2 analysis soon. Quality control. So we've recently started working quite intensively on metrics and some data sets for ensuring that you've got good quality coming out of your SARS-CoV-2 annotation. You might have noticed what the focus has been for a while. And then let me not forget to mention the training and workforce development working group, and then the ethics and data sharing working group, which complement what we're doing in the more technical work. Oh, and I nearly forgot the very important data standards working group that has set up a proposed metadata scheme for SARS-CoV-2, which I believe CanCOGen is using. Anyway, enough from me. Let me hand over to Christina.
Well, thank you, Peter, and thanks for inviting me today. It's my pleasure to be here. Okay, if I could share my screen. Okay. Great. Well, thank you, Peter, for the introduction. Like Peter said, I was not familiar with phage for many years. My background is in bioinformatics and computational biology, and for most of my past couple of years, I've spent it mostly on cancer research. Recently, I've now expanded into cohort populations, but still virus and epidemiology or virology is something that I'm not familiar with, but now, of course, learning a lot more because of the current situation. But what I've realized is a lot of the way we manage data in cancer is very much applicable to virus data as well. So that's why we have been able to build the Canadian VirusSeq data pool in a very short period of time. So just to give you a little bit background, looking at the Canadian data that is currently in GSED, this is data as of April 1st. We did fairly well in terms of submitting data to GSED. However, unfortunately, a lot of the data is actually missing the metadata. And also there's a large time gap between the time that the data is collected and the data actually makes it to GSED database. So as you can see here, we also collect out data based on provinces. And so there's a very heterogeneous way of collecting the data. And here are the charts showing the date of sample collection. And you can see that some provinces submit all that data, that date to GSED, while some provinces would be missing that date. And that is also applies to other metadata fields as well. And because of what we have observed, there's motivation, and this is funded by Genome Canada, in order to get our data in a more unified way and also get it faster into the public domain. So we want to have a real-time dashboard in order to see the Canadian VirusSeq data. We want to see better harmonization of the metadata for sure. And we know that, as we have seen other countries doing, releasing the data is really key to rapid identification and also tracking the variants of concerns. UK obviously did a really great job there, and we know that we need to do the same. Also, one concern is sometimes the data use and acknowledgement policy is slightly different depending on where you get the data. So for the Canadian data, we want to get a very clear policy statement that everyone can agree in terms of data sharing and acknowledgement. So we want to create a collaboration platform. So because of the way that our health system is, there are a series of public health labs funded by the different provinces. And then there's what we consider ourselves an academic research community. And in the past, we haven't really got a lot of opportunities to cross over into each other's domains and to collaborate. But this pandemic really gave us the opportunity, but it also means a bit of challenging. It's kind of like figuring things out, for the first time. And then we want to enable the development of tools. As we see, there are a large number of great tools for analyzing the SARS-CoV-2 genomes. We really want to make it easy to be integrated with the data so that everybody can use the same tools without having to reinvent the wheel or having to build their own, even though it already exists. So this is the team. Guillaume Book is the lead PI. And Lincoln Stein is the head of adaptive oncology, who has also been building knowledge base over the many years. We have Fiona and William from Simon Fraser. Yanzhou Li is a lawyer who is developing the governance and many of the policies for the CanCOGen VirusSeq portal. And Mark Fume is a DNA Stack that is a commercial partner to also host some of the analytics for the SARS-CoV-2 genomes. So this is the vision we have. This is the public health units who are doing the sample collection and the sample sequencing. When they have the data, some of it is already sent to what we call the National Microbiology Lab. This data they send to the NML actually includes sensitive data. So these will have identifiable data in there. So that's why it's controlled. For those who submit directly to NML, we're going to streamline the process so NML can submit the non-sensitive data to the VirusSeq portal. So anyone coming to the VirusSeq portal to view the data, to download the data, they do not need to have any login. They do not need to register. In some cases, the data may come to the VirusSeq portal first. Then it will be up to our job to actually submit to other public databases such as GSED or NCBI, or in some locations back to the public health database. So the idea is we want to really streamline it for the public health units because we know they're busy already. They have been doing a lot during this pandemic. We want to streamline in such a way that they will be very willing to share the data without being overburdened. So when I mentioned non-sensitive metadata, we try to keep it to a very minimal, and you probably are familiar with this because, as Peter said, this was the standards developed by PHA4GE, and then we adopted some of this. And Emma Griffiths is, leading the effort of the data harmonizer that helps users validate the data before submission. So we're really adopting the standards already formed, so we did not reinvent the wheel here. So to build this portal, there are a number of assumptions and requirements. As I said, we want to have non-sensitive data. We really want to lower the barrier of accessing data, but we do want to have all the metadata fields completed. Having those fields saying not provided, not available is really not useful to researchers. We want to first take on consensus sequences, and later on, if we take the raw sequences, they would definitely have to be dehosted, and there would be a process to make the validation. One requirement is that your portal must be easy to use. Like I said, for data upload, people are already familiar with GSET submission or to other portals where they can drag and drop or easily upload a couple files from their computer. We want to make it easy. Data search needs to be intuitive. That's actually easy to do because everyone is familiar with online shopping, and so this is easy. Data download. So obviously, there will be two kind of users who want to download all the data at once. It doesn't matter what version it is, they just want all the data at that moment in time. Other users want to just do incremental downloads. They don't want to have to download everything again and just say, hey, I've downloaded data last week already, just give me the difference from that point to today. And users will want to do this both through a web interface and some would want to do it programmatically using APIs. And as I said, we want a real-time dashboard. So we don't want the data to be submitted to us and have to sit there for a day or two before it gets into the portal. We really want to have almost immediate release of the data from the point of submission to being available in the portal. Yes, we need to streamline this. So as I explained, either the data can be submitted directly to the portal or to the National Microbiology Lab, and then we will handle the further submission to GSET. The major requirement for us is to build this portal in 4 weeks. And we were not talking about a prototype portal, we were talking about a production portal. So it was quite stressful during that time because our team has never accomplished this timeline before. Typically, it takes us 2-3 months to build something, even without the available technology and experience. So how exactly do we do it? First of all, we leverage our experience. So the genome informatics team at OICR has been developing data portals for many years. My predecessor was Vincent Ferretti, who's now at University of Montreal. So he has led the team building these portals for the International Cancer Genome Consortium. And this is a portal that has been evolving over past 7 years with growing amount of data. So it was really scalable and really stand the test of time. And we have also done other additional portals for the we've done portals for the National Cancer Institute in the US. Again, these are mostly cancer focused. Our recent portal for the International 100K plus cohorts consortium is primarily for population cohorts around the world. So if people are interested in finding cohorts that collect, say, blood pressure measurements, they can go to the Atlas and find those cohorts from around the world. And then our latest technology is actually the continuation of ICGC. This effort is now called ARGO, Accelerating Research in Genomic Oncology. This is where we can have a lot of freedom trying the latest technology and building a much involved system. This is going to deal with data submission from a clinical aspect, from the genomic aspect. We're going to run workflows on these genomic data. So for calling somatic mutations in cancer types, and then making the data available to the submitters to actually QC. And once the QC is done, the data is released to public for access. So this is a platform that will be developed over the next couple of years. We do leverage our existing software. Because of these many years of building portals, we recognize that there are some core functionalities that are just needed in every single portal. So we actually have refactored our code to make sure that these modules can be reusable when we go to start a new project. So these core modules are for authorization, for doing file transfer and storage, doing metadata management, of course, indexing the metadata so it's searchable. And so we have a search API that can easily interact with the index, which we use as our Elasticsearch index. And then the data portal UI would be generated very quickly in a generic way. And then we would spend further time to customize it. We do use third-party technologies such as Elasticsearch and Apache Kafka. So Kafka is something we recently used to allow us to basically listen to events, so that when an event happens, we would trigger downstream actions based on that event. So using our technology, we also have to have the compute infrastructure, because when we talk about a production portal, that means we got to have proper backups, proper monitoring. So we leverage a compute cloud that we have built here in Toronto. It's called the Cancer Genome Collaboratory. It's OpenStack, and so we already have a Kubernetes cluster set up. This really makes it easy for us to do deployment of Docker containers. So that's why we managed to get all the software deployed very quickly. We already have system monitoring going on, and data backups. We do it on a nightly basis, and we archive it to an offsite location. Obviously, with the VirusSeq data, it's very small, so it is actually pretty easy to deal with these kind of backups. I do want to point out some differences when building portals for cancer versus SARS-CoV-2. Typically in cancer genomes, we get usually sequencing reads, either in FASTQ format, or sometimes they're aligned, so in the form of BAM or CRAM. But here for VirusSeq, we start with just FASTA. Obviously, the sizes are a huge difference, hundreds of gigabytes for cancer genomes versus 30 kilobytes for a consensus sequence. So for the bioinformaticians who typically deal with cancer genomes, they're familiar with command line tools. They use scripts to do this. They typically pull a lot of the metadata from their existing laboratory information management systems. But here with SARS-CoV-2, I think there could be a range of ways people want to submit data. The easiest, like G said, is basically a drag and drop web interface. Some still prefer command lines using APIs to do scripting. And the metadata format is also very different. Typically, we have a JSON per sample to describe the metadata. And because the metadata can be quite complex, sometimes it's nested or in arrays. So that's why JSON is appropriate for this. But obviously with VirusSeq, one TSV containing many samples with about 15 to 20 columns is sufficient to submit the metadata. So much simpler. For metadata validation, we typically have a data dictionary. So there are two types of metadata validation in cancer. One is the clinical data that comes with the cancer genomes, describing the type of cancer, the type of cancers, maybe the stage of the cancer, etc. But there's also metadata about the genomic data, because we do need to know, for example, the number of read groups, the read group IDs, before we can do further down alignment and analysis. So there's definitely a lot more metadata to talk about. But here with the VirusSeq, great thing that we already have the standard developed by PHA4GE, and then you can use the data harmonizer to do the validation. When we store this metadata, though, we still stick with the JSON schema because that's our system already has that format. So for file transfer, for cancer genomes, yes, we can do multi-part, we have to do multi-part transfer. Basically, a file is split to multiple parts, and it can be transferred in parallel depending on the number of CPU gives it. And it still will take minutes or hours. But here, SARS-CoV-2 genomes, it only takes seconds. It's pretty easy. For data download, again, with the genomic data so large, is usually command tools for cancer. But of course, for SARS-CoV-2, these prefer to be downloaded directly from the web browser, so you can actually get at both TSV and FASTA very quickly. So for the development effort, we do need to establish some new microservices for SARS-CoV-2 genomes in order to do the data submission and also the download. So here's the architecture that we did for the VirusSeq portal. And as I said, submitters do require login, but downloaders do not. So we've built this out using many of our existing modules from our Overture software suite. I kind of named these before in blue. And then for this, the submitters do need to log in, but in this case, they want to use the institutional emails. They don't want to use something that like Google or Orchid, and that's why we use a Keycloak as a third-party software to enable the institutional emails login. So the service we specifically developed for VirusSeq in order to the upload download is called Muse. So this is basically the Swagger document showing you the endpoints of Muse. So doing download of a single FASTA file or in gzip format. We also show the submissions history for a user. And then we also show the have the endpoints to do uploads as well. So this is actually our VirusSeq data portal. This you can see on production. I really should have put in a URL here. Is actually virusseq-dataportal.ca. There's not a lot of data in there right now, and I can explain to you why. But for now, I'm just going to show you very quickly how to do a data submission. So I am here in the portal now. This is not an URL that you'll have access to because this is our development environment. And you see that we've actually submitted a lot of files during our testing just to repeatedly test ourselves. But it actually is the same number of genomes, just 4,000 genomes. With all these files. So in here, the reason I'm using this development environment is because I don't actually want to pollute the production data, but I'm going to show you how we actually do a submission. So here it is where I have a submission interface where I get to upload some files. And so I have prepared this already. So actually, I'll show you here. This is actually my dashboard with a submission that I did just yesterday with the date of June 1, and it has 38 viral genomes in there. So now I'm going to upload a different set of files. So if I just put in one file, this .fasta, great, but it's going to complain that I need actually one metadata TSV and one FASTA. So this is where we do some validation. We look for two files, a TSV and a FASTA, and then we also do a little bit of validation between these two files. So let me upload the TSV as well. So now I can submit. And so it actually validates that I have the same number of data in there and allows me to validate that the IDs are matching between these two files. Now, I'm not sure why. Let me try to upload a different set of files then. Okay, so it's one of those situations where when you're trying to do a live demo and it's not working. Okay. Let me go back to the portal here. So you can see that actually there's 17638 files in there. And if it is actually uploading live, this actually number should be increasing because what happens is, okay, so I've been locked out. That's probably why. Okay, sorry about that. Let me try that again very quickly. Okay, there we go. So basically the validation passed. So now I have 42 genomes. They're all queued up. And it's basically doing some very basic validation. Like I said before, originally, so you now see them being processed. In this portal, it was 17638, but as I refresh it, you see that one file has got added. So this is really real-time ingestion of the data into the portal. As soon as it's complete, it's immediately available. So this will basically go through, the queue, processing complete, and then making the data available in the portal immediately. So I'll just go back to here in the interest of time. The search that we can do is very easy. Like I said, if you know you've gone shopping, then you know the check boxes to narrow down your criteria. And when you get to the search, finish the search, you can download just the data that you satisfy your search criteria, so you don't have to download all the data all the time. So just quickly summarize some of the challenges we encountered so far. So at the initial launch, we had over 4,000 genomes from Quebec and about 100 from Ontario. They're all previously submitted to GSED before, but then we had a user reporting that the Quebec sample may have acronyms that potentially identify the patient if the number of cases in a rural hospital is low. So out of precaution, in the portal immediately hid the ID column. We also hid the function to download, since the ID is also in the FASTA file. But after some deliberation, I was just told last night that, okay, the Quebec data can actually be submitted to the portal as it is. They're not going to change the ID. It's fine. But the new data moving forward will use new IDs. So it is a lesson we learned, because it seems like we need to get a quick look of the data before it goes into the portal. Even though mechanically we can't release the data immediately, the curation team would prefer to have a quick look at the data. So we're going to change our data ingestion process a little bit. As part of onboarding, we're going to ask submitters to send a sample of the metadata to the curation team to take a look. And some groups actually prefer to just have the curation team always look at the metadata before submission. So there are a couple of things in progress. We are now refactoring our download API to meet more use cases. This API is new for VirusSeq, and as we now have to have more conversation with users, we're going to change it a little bit. We're going to have an acknowledgement page that is dynamically generated based on the data. So we don't have to manually edit it as the data comes in. Obviously, we haven't worked a lot on the documentation for submitters and the curation team, and that's what we're going to work on next. At the other spectrum, we've been engaging with the public health units. We want to understand their current data flow, the pain points. We want to make it as easy as possible for them to submit the data. We want to have a very good understanding for everyone in terms of data governance and policies, so we can encourage everybody to share the data. And of course, we want to agree on the minimal non-sensitive data so that everyone will be willing to submit those metadata without missing things. So lastly, I'd like to thank my team. These folks have been working really hard during this time. And lastly, here's our funder. Thank you for their support, and thank you for your attention.
Thank you so much. That was, wow, that was really rich. I see that we had a technical problem with our Q&A interface, but I do have a question here from Alan in the chat that he's asking, so what range of people are needed to support VirusSeq? So besides the devs, do you have curators? How does that work?
So for the technical side of things, because we had to build this so fast, I actually had 10 people on my team. And it actually is a composition of software developers, DevOps, a business analyst, and a bioinformatician. So that's just on the technical side. On the Simon Fraser team, they have 3 curators. So they will be the ones helping users to submit data and answer questions, anything regarding data, they'll be handling that.
And I was typing a question, but I couldn't type fast enough. But so this is from the kind of publicizing the data to the world perspective, right? But so from your earlier diagram, the different labs have access to obviously their own data, but is anybody using this as an ingest system as well, that you can basically draw data from VirusSeq into your analysis pipelines? Or is it mostly for non-Canadians that you imagine this being used?
Yeah, so we're actually going to integrate more with other downstream analysis pipelines and analytics and dashboards as well. If you're familiar with the visualization tool CoV2, that's another visualization tool we're integrating with. So this portal is going to mainly have Canadian data, but I can totally see a downstream platform that is outside of this, but basically ingest or put together more data from around the world.
Sorry, I've just seen another question come in.
Yeah.
Do you get requests from epidemiologists at national or provincial public health institutes for analytics support? So that means those people will often have richer, like you showed in the earlier diagram, metadata than what's actually submitted. So have you been, looking into how to support them? You mentioned dashboards like CoV2, but, what about when somebody wants to integrate with a wider range of metadata? Have you thought about maybe, giving authorized users access to richer metadata or anything like that?
Yeah, so currently the flow is that if users want to get more metadata, they should contact the data owner directly. And also, there's a host seek data portal as well that is being developed by another team. And in that case, they would be expected to host data that is under control access, right? So that may be a better place to have the control access data, because this portal is really intended for non-sensitive, completely open access data.
Thank you very much. I'm just seeing one more thing popping up. Yes. Are you concerned about cybersecurity at all for your portal? Obviously, with the data, it's not so much of an issue, but you still do have a system that needs to be protected.
Yes, absolutely. Especially our past experience, primarily with cancer human genomes, that is definitely PHI. So yes, cybersecurity is very much always on my mind. Some of our systems, first of all, that's why we have DevOps specialties, is we really control what can come into the network and what can go out of the network. And also we even think about what if someone comes in and actually edits your data? They change your data. They're not stealing it, but they change it. So we even have mechanisms in place to make sure that we actually keep an eye on our own data to check nothing has deviated too much, even between submissions.
Yeah. Okay. Yeah. So I've got a couple more questions from Danny and Alan. I'm just going to ask Danny's question, but we'll have to leave Alan's one for any time we've got at the end. So could the DNA, could the data feed into something like DNA Stack or similar?
Yes, in fact, yes. So DNA Stack has developed the COVID cloud, and I'm actually part of that effort as well. So we will be integrating the data to DNA Stack. So in one of my slides, I show you that we have a Kafka event queue. So basically, we'll give DNA Stack access to the queue. That means they can listen to the event as well. As soon as data gets submitted, they can provide the actions they want to either download the data or swoop it up to update the dashboard. So we'll try to integrate in a very seamless way.
Okay. Thank you so much. As you can see, there are more questions, but time is moving on. And yeah, so I think we need to hand the screen over to Sam Nichols, Dr. Sam Nichols from University of Birmingham, who will be talking about Majora and its use in the UK COG. Sam, welcome.
Hi, thank you. Just share my screen with you.
Yep.
Great, thanks very much for the introduction, and I really enjoyed Christina's talk. It's going to be a sort of similar story from us in the UK. So for those of you who don't know who I am, you might know me from Twitter. I'm a fairly outspoken bioinformatician, previously worked on reconstructing strains out of stool, but that is a previous life now, and as you're all probably aware, we're in the midst of a global pandemic. This is a chart that I took off the UK government's website this morning. On the x-axis is time, on the y-axis is positive SARS-CoV-2 cases in the country. We've had our first wave, we've had our second wave, we've had our third wave. We're on track for a fourth wave, who knows? But I think this really speaks to our kind of lack of pandemic preparedness in the UK, which is a bit of a surprise because pandemics aren't new. We've been surrounded by pathogens and pandemics for a very, very long time. And so this paper that I'm highlighting here is one that you're probably all very familiar with, and I'm probably preaching to the choir about why genomes are so useful for looking at the spread of pathogens. But if you're not, if you imagine the idea of taking an isolate of a pathogen out in a community where you know it's spreading, and you DNA sequence it, and you do this for a whole bunch of people suffering with this virus in the community, you can look at all those genomes and you can look for differences in them that tell you about how that virus is moving through your population. And that's what this paper is talking about. It highlights various ways that this has been used in the past for a good public health intervention. And that's what we wanted to do in the UK. So you might be aware that last March, we founded the Covid19 Genomics UK Consortium, lovingly known as COG UK. It's a national network of countless NHS organisations across the country, all 4 of the UK public health agencies, some 20 different academic partners, the Wellcome Sanger Institute, and these lighthouse laboratories that I'll talk about a bit later. These are mega high throughput testing laboratories set up by the government. And this was all set up with £20 million worth of funding from the UK Department of Health and Social Care and the UKRI and the Wellcome Sanger Institute as well. So the 3 main goals of COG UK was to produce data by connecting a network of sequencing capable organizations to a network of sample collecting organizations. And then we want to collect all of that data and provide some sort of unified system to transfer and store the consensus genomes and metadata for all of those samples, and then integrate that into a single data set for people to use by harmonizing all of that stuff and making it available downstream for analysis. So there's not a lot to say on producing beyond what I've already said. You can imagine a sampling network of GP surgeries or hospitals or anywhere that's collecting samples from out in the community. And those come through to local sequencing sites. And what I've highlighted here, the most important aspect of this is that those sites that participate in COG are responsible for choosing how they want to do their extraction and library preparation, their sequencing, their bioinformatics. So they're all autonomous of the system. And this really reduces the burden on participating and also allows for us to use the expertise of each particular site rather than enforcing a particular protocol on everyone. So all of those sites generate a consensus and an alignment for every sample they sequence. And then on the right-hand side of my chart here, some poor person at that site is responsible for taking in all of the different sample metadata, managing it and merging it into some consistent document that they can share with us. And I'll talk about that a bit later. In terms of how samples come to us, there are two main routes, 2 pillars, we call them in the UK. On the left here is pillar 1. This covers NHS hospitals and public health laboratories. And on the right-hand side, pillar 2 covers community tests through those mega high throughput lighthouse labs I told you about. And they come into this pyramid. At the top of the pyramid, you've got low throughput, fast turnaround sites. So these are smaller NHS and academic laboratories. And on the bottom, you've got centralized sequencing facilities with high throughput, slow turnaround. Sorry. And the idea here is that you can explore both sides of the coin. So at the top, you've got smaller labs that are capable of looking at small localized outbreaks, and then the high throughput, slower turnaround, centralized sequencing facilities from large academic labs. And the Wellcome Sanger Institute are able to give you a broader idea of more samples from your population. So a broad backstroke idea of what the pandemic is looking like at a national scale. So now we have this data to collect. We want to bring it in together to the consortium. And I think this is where I come in. This is one of the most exciting parts of the project, this idea of decentralized sequencing, but it all needs to come together. And it's a real sort of interoperability problem. How do we get all of this data generated from various different sites in various different ways together in one place for it to be useful? So we need a location that can be reached by everyone in the consortium that has the capacity to hold the sequence and the metadata for a given sample. The sequence and metadata needs to adhere to some sort of specification that we apply evenly to every sample submitted by the consortium. And then putting all of that somewhere, say a database, should be as easy as we can make it. And ideally, we would like to be able to get it back out again. So here in Birmingham, we're very well placed in terms of location. We're a founding member of CLIME, the Cloud Infrastructure for Microbial Bioinformatics. It's an award-winning, high throughput system for doing microbial bioinformatics in the UK. It's hosted in various sites, but in particular, CanCOGen is a sort of secure slice of Climb that we're hosting in Birmingham, and it's mirrored in Cardiff. We have 0.5 petabyte of fast SSD storage. It's all in our state-of-the-art warm water-cooled compute data center, and everyone in COG has access to a shared 96 CPU, 3 terabyte shared login node, and they can farm out jobs to a variety of 40 CPU, 192 gigabyte compute nodes. So there's plenty of storage for samples, but we still need a home for the metadata itself. And before we can talk about where we store the metadata, we need to go back to that idea of a specification. As Christina was saying, that sequences have very little utility without any corresponding metadata. So we decided very early on that our analysis pipelines would just not process samples without it. And this was our sort of stick to make sure that we actually get the metadata for the samples that people want to upload to our system. It was obvious from the beginning because we're quite focused on viral phylogenetics. We want to know where and when the samples were collected so we can build phylogenetic trees. So it's a priority to collect that sort of information, but it was less obvious to us that some centers would actually struggle to find out when a sample was collected at all. Even seemingly random identifiers could be considered identifiable by someone. You may not know this, but England is separated into 6 metropolitan counties, 75 non-metropolitan counties, and London is set up in 32 different boroughs of its own. So ADM2 was even a difficulty for us to collect. So you might think, okay, well, perhaps outer postcode would give you a nice way to collect location, but this at the beginning was considered identifiable information. We had quite a long legal obstacle course to get the ethics signed off to be able to collect that piece of information. So we had many, many meetings on what to collect, but ultimately it was virtually impossible for any of these fields to be mandatory. And the true difficulty here lies in not generating a specification, but generating one that considers what can actually be extracted given the staff time and resources on the ground. So there's no one-off solution here. And because of that, we chose a very minimal specification. So we ask in COG for at least a unique sample name, the collection date, if it's available, or if you don't know that, the date that you know it was received in a laboratory, and the country it was collected in. Ideally, we want more than that, the county, better yet the outer postcode, perhaps patient age and sex, and sample CT values for helping to pick or look at QC information. So we have the specification. How do we collect it? Uploading metadata should be as easily as possible shouldn't involve sort of poking holes through firewalls. It shouldn't require any specialist applications to be installed, and it should be suitable for non-technical folk because we're going to be doing this across the country from all sorts of different laboratories. So dare I say it, at the intersection between intuitive and portable is spreadsheets. They're easy to fill in. Almost everyone has access to some sort of spreadsheet software, and they're very intuitive. So on the right-hand side here is just an example of what such a spreadsheet might look like. You've got a column for each of the metadata fields, and then each row represents a sample. So we've got a unique sample ID, a country, and a receive date here. So we have these CSVs. How do we collect, validate, and store them? We obviously can't rely on embedded software-based validators. It's not feasible for someone to collect and moderate these files manually. And ideally, we just don't want any humans to be involved in any part of this process because we want it to be quick as possible. So this is starting to sound like a job for computers. And so this is really where I came into COG. I think this is the system that I've built. I think it's fair to say that Majora is the brain of COG UK. It's a database that stores all of the COG metadata. It's also a website that shows limited pieces of that metadata to users and allows them to manage their accounts and different ways that the pipelines work for them. And it's also a set of APIs that allow the database to be updated or queried. So it's effectively a national scale LIMS and then some. It was built for COG from scratch with Django, which is a Python web development framework. We had our first registered user in just 3 days, and it unified the entire metadata process for COG in just 3 weeks. So I said API, Christina introduced these, but if you're not sure what I'm talking about, an API is an application programming interface. It's basically a way for a computer program to interface with a human or other computer programs. You can imagine it that you just send a structured message to a program over the internet, and it just gives you a message back, and that's an API. And so all interactions with Majora are done through machine readable APIs like this. And here's an example of some of those. So on the left-hand side here, this is how it's a JSON formatted API request that you would send to Majora to say, hey, I've got a new biosample. It was collected in England, collected in Birmingham. Here's a bunch of information about it. And then on the top right is Majora's response. And it says, I see your metadata. This all looks good. Here's the biosample ID. I've generated it and I've added it into my database. And this means it's available for everyone in the consortium to query straight away. So we're doing real-time validation here. And that means we can also throw out bad stuff before it even touches the database. And it also means that people know that they've passed this bad metadata without having to have some sort of human involved in telling them what's wrong. So if we tweak the collection date to be in the future, on the bottom right here, Majora replies and says, there's a problem with this particular sample. You can't collect the sample in the future. This is garbage. I'm going to ignore it and throw it out. So this is useful, and we've processed between May and March of this year, we processed 5 million of these API requests. That's 16,000 a day, and this is really how Majora works. Everything is done through these endpoints, but our users are humans, not machines. So how do we get these machine-readable APIs to communicate with humans? So luckily for me, Anthony Underwood at the Center for Genomic Pathogen Surveillance built the Metadata Uploader, which I've got some screenshots of here. So people are able to log in on the top left there. They log in with their Majora credentials, and then it presents a drag and drop interface. They can drop their CSVs into here, and then better yet, they submit them. And then this JavaScript front end processes Majora's response, and then it renders what's wrong with their metadata if there's anything to be corrected. These users then know straight away that they need to correct this and resubmit. So this is what that looks like as a sort of flowchart. On the left, you've got local sites building faster and BAM from each of their samples. They upload that to us through some secure copy or rsync method. The metadata is munged into a CSV, and then that goes through that uploader to be converted into JSON. And then on the right-hand side here in purple, it's loaded into Majora, and that is then responsible for loading the database with the artifacts and processes. And what are artifacts and processes? These are the core classes of metadata held by Majora. So an artifact is just a thing to be tracked, like a biological sample, a sequencing library, a reads FASTQ, or a consensus FASTA. And then processes are actions that happen to those artifacts. So a sample is collected, samples are pooled into a library, sequencing run generates a FASTQ file, a bioinformatics process generates a consensus FASTA, and so on. Majora records how each of these artifacts comes into existence and how all of those artifacts are linked together by processes. So we have a full audit trail of where a sample came from and what has been done with it the whole time that it's been on our system. And this is a screenshot of Majora. I'm not going to spend too long on here, but I just want to drill onto the right-hand side here. We have this downstream artifact section, and this is just what I've been talking about. You can see on the website, we have a biosample. It's been pulled into a library. It's been sequenced, and the sequencing run has been base called to generate some reads. And we have this for everything in the consortium. So going back to my flowchart back at the beginning, on the left-hand side here, we now have files copied to Climb. So we have a secure place to store uploaded sequences. And on the right-hand side, that merged metadata comes through our application programming interfaces, our APIs, and they're stored in Majora. So we have Climb for storage and Majora for metadata storage. It's now time to integrate this into a downstream data set. So if Majora is the brain of COG, then I think it's fair to say that ELAN is probably its heart. It's a Nextflow pipeline that's responsible for checking the consensus and alignment files that have been provided are both actually there and present and are valid, carries out a bunch of QC tests, and it adds those metrics into Majora. And it's also responsible for linking the QC data to downstream artifact directories for analysis to be able to pick up and use. So this scary looking flowchart is a sort of a way of me showing, I'll step you through this and show you how this works every single day. So local sites upload metadata to Majora on the right-hand side, and they securely copy their sequences to Climb, which is this little blue strip. ELAN then uses a piece of software called Ocarina in yellow here to find new samples. So it queries Majora to find new samples, and then it looks for them. It looks for the matching files on Climb. ELAN then processes those sequences. So it runs a bunch of quick checks. It runs a lot of SAM tools, and then it generates a bunch of downstream artifacts. And it's also responsible for telling Majora about what it's seen. So it adds all the files that it's encountered, adds all the QC metrics, and says, hey, can you group these things together? They're all part of the sort of record for this one sample. And then outbound pipelines are able to use Ocarina as well, and they can pick out of Majora things that they want to see. So we might want to package up high quality sequences for one database, such as GISAID, and then query for BAMs to be packaged up for ENA. And all of this is completely transparent. So ELAN's responsible for triggering other pipelines and also telling everyone about what has happened. So we have a fairly well-populated Slack instance for COG UK, and it sort of spews messages in constantly about what metadata is missing, what people need to look at, what's been found, and whether pipelines have completed successfully or not. And on the left here, back in September 2020, this is when we hit 50,000 sequences. And on the right here is the first message that was sent by ELAN to other pipelines to say, hey, I've completed. And underneath, you can see that Philopipe, the downstream phylogenetics pipeline, sees that message and replies and says, okay, I'm going to start as a result of that finishing. So all of these pipelines trigger each other. So the humans are not involved in this part of the process. And the nice thing about having the QC information in Majora as well is that you can actually query Majora straight away for data sets that match whatever QC you're looking for. So you might be interested in things that have failed QC for some sort of analysis about CT values, or you might want to get the stuff that has passed the highest level of QC. You can do all of that through Majora. So we've spent about 123 years of CPU time in the first year of CLIME-COVID with over 5.1 million jobs. But you might be thinking, okay, well, what does this enable? So the headline figure is today we've actually, this was yesterday, we hit 488,000 sequences, but today we've actually hit 500,000 SARS-CoV-2 sequences processed on CLIME-COVID in the UK. So obviously this is a huge milestone for us, sort of a half celebration in sort of admiration of what we've been able to build, but also a very somber milestone for us as a nation. And COG is also responsible for a whole host of other studies. You might see these on the website, the HOKI study, the genomic study, and so on. It's also responsible for generating a whole bunch of reports for SAGE, the public health agencies, the UK government. And I won't spend too long here, but it's also been used for countless analysis of hospital and care home outbreaks, workplace outbreaks. This particular paper from Science that I really like shows how the COG data set was used to look at how SARS-CoV-2 actually arrived in the UK, and was it through the way that we thought? And it was actually seeded from a lot of European travel. This might have been a surprise at the time. This is the sort of thing that we can look at now with a data set the size of ours. And you can look at all of this data right now on Microreact. So this is a web front end built by David Anderson's group at the Centre for Genomic Pathogen Surveillance. And you can go and look at everything that's circulating around the UK and explore the data set right now. And I'd be remiss if I didn't mention that, of course, it was the COG data that first picked up the variant of concern in the UK at the end of last year. And this is where some of our work has been going. We've been working quite closely with the public health agencies to help genomically confirm these case counts. And more recently, we're helping to solidify definitions of variants of concern and variants of interest for calling across the network. So to summarize, we've architected and deployed an end-to-end infrastructure to support distributed sequencing at a national scale. The infrastructure was built very quickly, but it was built by people with domain expertise. So I think even if we had to do all of this again, we'd make a lot of the same decisions the same way. Something I really want to highlight here is that prior investment in CLIME computing infrastructure was key in establishing COG. I just don't think it would have been the success that it is today if CLIME had not already existed. It was quite easy for us to take some resources out of CLIME to form CLIME-COVID, and that was because it was a well-funded existing infrastructure. Our use of APIs has enforced a standard interface for providing metadata, and it's also fostered an environment for development of tools against a common platform specification. And I can't really emphasize this enough, that everyone had to use the system in the same way. And that is why it was able to collect so much metadata, because there were no exceptions for someone processing metadata from one site compared to from somewhere else. Everyone had the same template, everyone had to use the same APIs. Our walled garden approach allowed for fast turnaround as well as high integrity. So rather than pushing everything to public databases and waiting for sessions to be generated and pulling it back down for analysis, and also risking contaminated metadata or anything going into public databases before we'd had a look at it, this system allowed for us to pre-process and QC check things that were coming in before we released it to the public. Within 1 week in March, we generated more genomes than China, and within 2 months, we were responsible for half the world's available SARS-CoV-2 sequences on GISAID. So it was a real step up generating because of this system. But all of our obstacles were not necessarily technical. Future systems like ours, I think, would do well to think about how to generate shareable sample and patient level identifiers, which was a bit of a problem for us at the beginning. I think COG will certainly serve as a framework for how to enable data sharing between different public health agencies and academic endeavors like ours in future. The last thing that I want to leave you on really is that bioinformatics is not just about cool algorithms. For the past year, I've been working on all sorts of different things like documentation, miscellaneous glue scripts that no one will ever see, API endpoints, ELAN itself. And all of these things are not necessarily big hitting bioinformatics pieces of software like a new aligner or a new assembler or so on, but all of these things are equally important. It's important to have good documentation, and glue code is the thing that actually makes everything run, right? But this wasn't just me alone. Everything that I've talked about today was built by us on CLIME-COVID, but there are countless other people who've worked on all different downstream bits of software, and we've sort of formed a ragtag group of bioinformaticians called Dippy. So I just want to sort of shout out to them, and there's plenty of talks and tweets talking about the sort of stuff that they've been working on too. So that's what I really want to say. A special thanks, I think, to Radislaw and Matt Bull in Birmingham and Cardiff. The three of us kind of really set this system up together, and thanks to Nick and Tom for giving us the free roam to build it in the way that we thought it should be done. Big thanks to Anthony Underwood for building the metadata uploader, so I didn't have to. And then to Michael and Ewan, who are really responsible for getting the ethics and sorting out the metadata specification early on in COG as well. So you can find out, there's obviously countless people to thank. There's like hundreds of people involved in COG. You can go and look at them all on the COG Consortium website. If you want to hear more about what we've done, we were actually recorded a little while ago on the MicroBinfy podcast. And if you want to read about what we've done, we've got a preprint on bioRxiv as well. But other than that, thank you very much for listening.
And thank you very much for that speed run through Majora and COG and how it all works. And that was really amazing. I mean, both talks today have been absolutely amazing to me. I've got a few questions here. The one is a two-part question, actually three-part, but I'll summarize into two parts from Soyeon Kim, where she's asking about the data models, whether they would integrate with other sources of data. Have you used existing data models like PHA4GE? And then what are you using for authentication when people are uploading? So that question first, yeah.
So for data models, we picked our own data model. So we've built it to be as flexible and as customizable as possible. And I could probably talk for quite some time about whether that was the right or wrong thing to do, but that's what we did. For authentication, originally, we generated private passwords for people to use with the metadata uploader, for example, like a service key, but everything runs on OAuth now. So that's an industry standard authentication platform.
And then I've got a kind of question that kind of follows up to this from Alan Christoffels, that do you allow like upload of, for instance, barcode IDs, so that, for instance, if somebody's got a LIMS in the lab where they're scanning in all the samples, and then you could directly interface to Majora?
So we don't have anything like that at the moment, but people can provide something that I didn't talk about, is people can provide their local laboratory IDs that went into the sort of public health agency system. And those are then actually securely passed through to the public health agencies in the UK, who are able to pull the linkage down for their own analysis as well. So the public health agencies are actually able to use CanCOGen to link to their own richer metadata that isn't shared with us, because it's obviously quite restricted.
Okay, so that's a bit like what Christina Young was talking about, where you have a partition of nobody else can see this with metadata, and then you've got the stuff that basically ends up in your publications, or I mean in your public-facing things. Yeah, and then actually I had a question for Christina, because Sam mentioned Climb and its role in this. The cloud that you're using for the VirusSeq, was that built, special purpose built for, during the SARS-CoV-2 pandemic, or was that a pre-existing cloud facility?
It's a pre-existing cloud facility. We built this many years ago and have been using it for cancer and many other projects. So we kind of piggyback on it. Otherwise, we won't be able to get it up and running so quickly.
Yeah, I was wondering about that. And then back to Sam, you mentioned Cardiff and I think Birmingham as the two sites that you're using for Climb. How has it been to work in a kind of distributed or kind of federated infrastructure like that? How are you moving things between different bits of storage and compute and so on?
So it's been quite challenging. The main thing that we're having that we had to solve at the beginning was how we replicate the data on the other side. And at the moment, it's sort of a network of rsync commands which seem to be holding up for the time being. But we do want to look at, so at the moment we're running Birmingham as sort of a primary site, and Cardiff is a more of a fallback site. And also we do a bunch of different analyses in Cardiff as well. But I think we want to look at more of a sort of active model where both sites are able to do things at the same time and that sort of thing. But that's a whole different thing that I could probably talk about.
And with that, I think we are officially out of time. Jamie, does that mean that the webinar will just close, or can I abuse time for just a couple? Oops, it will apparently automatically close soon. I had one question to think about for the follow-up, which is Climb is using more of a kind of traditional POSIX file system storage model, right?
Yeah.
Yeah, it is. And then just to confirm that VirusSeq is more organized around object storage.
Yeah, we're definitely on object storage. Yeah.
Okay, yeah. So no, it's something that we've been discussing in the infrastructure working group. For those who are interested in the infrastructure underneath bioinformatics, as Sam said, this is not all fancy algorithms. Then we have an infrastructure working group where one of the things we've been talking about is different challenges in data storage, whether you are running on-premises infrastructure or infrastructure as a service, as some people are doing, or even the kind of platform as a service offerings that are out there at the moment. So yeah, feel free to look us up on phage.org. That's PHA4GE.org, and perhaps join in our discussions. As for the rest, thank you everybody for joining. Thank you so much to the two speakers. I do not have chocolates or anything like that to hand out to anybody, but it has really been amazing. And I'm sure that we might email you with follow-up questions in the future. And see you all next time we have a webinar. Bye.
Thank you.
Thank you. Bye.